Okay, so these next slides over here that I'm going to show you is basically a reiteration of the stuff. We're going to look at it closer with pictures so you can see things. Now, one thing I have to tell you about the mitochondria, because it does take place in the mitochondria, but the mitochondria, as we've told you before, it has two different membranes on it. I'll just draw it very quickly here and I'll erase it. But you have an outer membrane, all right, so the outer membrane there that kind of keeps it away from the cytoplasm, right, divides it. That's basically what this is. But inside there's another membrane, and it's undulated. So we have an outer membrane and an inner membrane. The inner membrane is undulated. But we also have spaces in between there, right? We got a space in there, and we got a space in there. The space inside is called the matrix. The space in between the inner and outer membranes is called the intermembrane space. Where the citric acid cycle takes place is in the matrix. The electron transport chain actually happens, well, it's these molecules that are embedded into the inner membrane. So it takes place, the hydrogens will be pumped out of here because it's coming from the TCA, pumped out of here into the intermembrane space, and then it has to go back in. But there's a machine over here, an ATPase pump, ATP synthase, that is going to make the ATP. But what makes that run is all the hydrogens that are going out here, they go back in, and every time one goes in, I'll show you a little video, it kind of cranks it to produce an ATP. We're using the power of these hydrogens, we're using the power of these hydrogen electrons to make ATP. So the three stages, just to reiterate, the three stages of cellular respiration is glycolysis, which happens in the cytoplasm. It's going to break this 6-carbon structure into two molecules of 3 carbons. And they're called pyruvic acid or pyruvate. Yeah.
So what pumps it out? Like you said,
Where?
it goes from a matrix.
Yeah, we'll get into that. We'll get into that. I just want to give you an overview of this, and then we'll spend more time talking about the electron transport chain. I did do that right. Right? I did do that right. Electron transport chain. Then we have the citric acid cycle, Kreb cycle, same thing. And that's going to complete the breakdown of the original glucose, right? It's going to take this pyruvic acid, you got the acetyl-CoA, and then it's going to enter here, and we're going to produce a lot of different things here. Then we have something called oxidative phosphorylation. We're going to put phosphates onto something. And oxygen is going to be used for this. Oxidative phosphorylation. It occurs in the electron transport chain. This makes 90% of all the ATP. The other 10% is made by here, as you can see here, and also made here by the GTP. this to occur, you need oxygen. And the electron transport chain is powered by redox reactions, which is why I talked about that yesterday. So, showing the same thing, just in picture form. This is the cytoplasm, or cytosol, right? Here's your mitochondria. Kind of shows you the two membranes over there. Glycolysis is happening over here. Take the glucose, make the pyruvate. Say pyruvate, pyruvic acid. Same thing, for your purposes. This is a process called glycolysis that occurs in cytosol. You are going to make ATP. Not lying about that. You're also going to make a couple of NADHs. That's over here. Now the pyruvate, if there's oxygen, can enter the mitochondria. When it does, we're going to have the Kreb cycle go on, the citric acid cycle. You're going to make ATP from there, or in this case, GTP. But it's also going to make a couple carbon oxides. It's going to make a lot of NADHs, FADH2s. These guys are carrying those hydrogens that are going to make more ATPs. The electron transport chain is happening here. All right? It really happens in the membrane itself, the inner membrane. I'll show you that. But that's where it's going to convert these FADHs, H2s, and NADHs and turn them into ATP. Again, for every one NADH, you get 3 ATPs. For every one FADH2, you get 2 ATPs. So look at what's happening here. Follow the numbers. How many FADHs make, how many ATPs do you get from there? 2. How many do you get from one ADH and NADH? 3. But we're multiplying by 3, so you get 9. So we have 9 plus 2 over here, plus 1 more over here. What number are we up to? 12, right? Am I doing it right? So 9 plus, 2, that's 12. All right? Net-wise, you get 2 over here. That's 14. Does that make sense? Well, before I do that, so we have 12 over here, but for one molecule of glucose, how many do we get? Not 12, but 24, right? Because I was talking about one molecule of pyruvic acid coming in. Are you following it?
Yeah.
Now you multiply it by 2. Meanwhile, you got another 3 here, but 6. Because there's 3 of them that come from 2 of those. You see how we're following it? It's already up to like 30.
Yeah.
Then another 2 over here. You got 36. You see what I mean?
I thought we were having 1 in that one.
But you have 2. Like I said, 1 pyruvic acid coming in here, but from one molecule of glucose, you get 2 of these, which means you're going to get 2 of these. You got to follow that. This uses oxygen. You're not going to, if you don't have oxygen, you can't even convert this to energy, to ATP. So you're getting roughly about 38, really 36 net ATPs with oxygen. from 1 molecule of glucose. You make 38, but net-wise, you got to give back 2 over here. But if you had no oxygen, net-wise, how many ATP are you going to make? Total. From 1 molecule of glucose, net-wise, how many ATP do you make without oxygen?
4.
Net. 2. Look. Without oxygen, oxygen is only used here. This is not, like I said, look, anaerobic. So this does not use oxygen. You can go from 2 and make 4. Net-wise, you got to give back these 2, so you only make 2.
How about the ones at the bottom?
These can't get in there. You need oxygen.
So it won't.
It can't. You need oxygen to do this. You see? Don't memorize. Understand where they're coming from. I can ask questions like, if I had 3 rounds of acetyl-CoAs coming in here, how many carbon dioxides are made? 6. Yeah. You got to understand the concept. Certain things you got to memorize, how many at first, but then the concept's pretty easy. So as I said, I want you to know what goes into each process, what comes out of each process, how many ATPs are actually made, and where do they occur? To emphasize that you need oxygen. All right, so let's look a little bit, let's reinforce the glycolysis procedure. So it occurs in cytoplasm, uses no oxygen. All right, we're just talking about this area right here, right? So it's anaerobic. It's a series, like I said, 10 different reactions. Don't worry about all those reactions. But it basically does this. The big picture. It takes a glucose molecule, 6 carbons, and turns them into 2 pyruvic acids, or pyruvates. There's one, and there's another. I kind of did it color-coded for you. All right? I only showed 1 up here, but we're multiplying by 2. Otherwise, the picture gets really busy. We're following? Right? There's an investment phase going on with this. We need to use 2 ATPs to get that running. But the payoff is that you get 4 ATPs. So you make net 2 ATPs. And that's done without oxygen. You're not going to get very far with that. Hold your breath and find out how long you can last. Hold your breath and run a marathon and see how long you can last, right? Also, 2 NAD pluses get reduced to 2 NADHs, which we will use later if we have oxygen. If not, they'll go right back. We'll talk about that too. Same thing. Glucose molecule here, 6 carbons. Glycolysis will break it up into 2 pyruvic acids. Oh, here, 2 ATPs are needed, but you get 4 out of it. Net-wise, you only make 2. Also, which doesn't show on here, the NAD pluses over here. We have 2 of them that are made, that are used later. So my pictures are up there. That's what I'm saying. I wish you had the video. step by step. Like I said, the colors might be a little bit different, but pictures are there. Questions on glycolysis. Again, if there's no oxygen, this we can use. We can change it to something else. We can't change it into energy, but it gets turned into something else. And you've all felt what it makes. Lactic acid. It makes you tired when you run, and it burns and hurts. Okay, questions so far? All right, now, if there is oxygen, then this pyruvic acid has a path to go backstage. It has to, it's going to go into this citric acid cycle, but it has to prepare that pyruvic acid to enter it. A 3-carbon structure can't enter here, only a 2-carbon structure. Now, in eukaryotes, this occurs in a mitochondria. A prokaryotic cell, it occurs in a cytoplasm. There is no mitochondria there. So we have to oxidize pyruvic acid. This requires oxygen to enter the mitochondria. So this is an aerobic procedure. The pyruvic acid changes to acetyl-CoA. And this is an important step to enter into the citric acid cycle. We want that citric acid cycle to work, because this is where we're going to get all that energy. That's the big picture. When this happens, carbon dioxide comes off. Why 2, and I'm only showing one here, because like I said, I'm only talking about one pyruvic acid entering here. But with one molecule of glucose, you get 2 of these, which means you get 2 of these. You've got to follow the numbers. So the 2 pyruvic acids, which are 3-carbon structures, become 2-carbon structures as called as acetyl-CoA. The carbon dioxide gets released. There's the other carbon that was attached to the in the pyruvic acid. Also, 2 NADHs or 2 NAD pluses get reduced to 2 NADHs, which are shown right here. Again, I'm only showing one, because I'm only talking about one pyruvic acid. So there's a total of 2 for glucose molecules. Be careful in my exams when I say, what do you get from one glucose molecule? Whatever numbers you get, you got to multiply by 2 at those proper places. So showing the same thing over here. Pyruvic acid, when it enters the mitochondria, carbon dioxide comes off. All right? Don't worry so much about the steps in between, but there is something that's called a coenzyme A. It's added to this structure. Now, that's why it's called acetyl-CoA. NADH comes off of that, and now you have this acetyl-CoA, which is a 2-carbon structure. Carbon dioxide is where that extra carbon came off. And my picture here. And I kind of summarize things over here. 1 acetyl-CoA, 1 carbon dioxide, 1 NADH created by 1, and that's why I emphasize that 1 pyruvic acid molecule, not per glucose. Good so far? Okay. All right, now the citric acid cycle, or Kreb cycle, which is here. This occurs in mitochondria, eukaryotes, and in the matrix. So within the space, that bigger space that's in the mitochondria. This you need oxygen for. And there's 8 steps with different enzymes. You don't have to worry about those 8 different steps. Okay? And you don't have to worry about all those enzymes. Okay? Thing over here that's going to happen is that you're going to get that acetyl-CoA, 2-carbon structure, with oxaloacetate, which is a 4-carbon structure. It'll make something called CITRIC. That's what I call the citric acid cycle. But it's one of those structures all in there. There's 7 other steps that go on in this whole thing and bring it back to make oxaloacetate again. So that it'll take another acetyl-CoA and do it again. And a cycle, and a cycle, and a cycle, and a cycle. One cycle, using 1 acetyl-CoA, you'll get 1 ATP, or the GTP as I showed you over there. You get 3 NADHs, and you'll get 1 FADH. It'll also release 2 carbon dioxides you can see over here. I kind of put this up here just to show you that I'm very kind to you guys. All right? College, when I took biochemistry, you had to know all these names of these over here, each enzyme that occurs between each one of these things. You have to draw out each one of these things. Oh. Merry Christmas, Happy Hanukkah to all you atheists. I don't know what to tell you. All right? You can simplify things a little bit too. But understand the big picture here. So you have the acetyl-CoA over here and the oxaloacetate over here entering this citric acid cycle, Kreb cycle. When it does, it creates all those things. All right. So for every 1 acetyl-CoA molecule that enters the citric acid cycle, you get 2 carbon dioxides, 3 NADHs, 1 FADH2, and 1 GTP or ATP. For 1 molecule of glucose, you multiply that by 2.